Hello, everyone, and welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. I'm Amar Gujar. I co-lead the Neurobiology and Brain Tumor program with Susie Baker. And it's my real pleasure today to welcome Dr. Paul Northcott from St. Jude to present the seminar. But before we start the seminar, I'd like to make some housekeeping announcements. I'd like to briefly mention that you can submit the questions at any time during the talk using the chat function and sending them to all panelists, or you can email them to socc@stjude.org. If you are experiencing any technical issues during the talk, please contact SOCC support@stjude.org. Dr. Northcott will leave time for questions at the top of the hour and has also kindly offered to stay on for some additional time after the close of the official event if anyone would like to stay on for further discussion. And now I'm really pleased to introduce Dr. Paul Northcott. So Paul, it's a great honor and pleasure of mine to introduce you for this seminar series. You are a friend, colleague, and even a mentor to me. Dr. Paul Northcott is an associate member in the Department of Developmental Neurobiology at St. Jude Children's Research Hospital. During his PhD and postdoctoral training at the two best Medulloblastomics labs in the world, in Toronto and at DKFZ, Paul made several seminal contributions to the medulloblastoma literature, including the description of molecularly and clinically distinct consensus subgroups, comprehensive annotation of the genomic and epigenomic landscape, and the discovery of the enhanced hijacking as a novel mechanism of oncogene activation in brain tumors. Paul is an active member of the Children's Oncology Group, the American Association of Cancer Research, and the Society of Neuro-Oncology. And he serves on the scientific advisory board of the Brain Tumor Charity in the UK. He has led several seminal studies which are published in Nature, Nature Genetics, Lancet Oncology, and the Journal of Clinical Oncology, disclosing the molecular mechanisms and underlying medulloblastoma pathogenesis through high-resolution genomics. He is the recipient of numerous prestigious awards, both as a trainee and as a principal investigator, including the Stuart Allen Hoffman Memorial Prize, the DKFZ Alumni Prize, the V Foundation V Scholar Award, the Sontag Foundation Distinguished Scientist Award, the Pew Stewart Scholar Award for Cancer Research, and most recently, the prestigious St. Baldrick's Foundation Robert J. Arcisi Innovation Award. He was also recently awarded the inaugural recipient of the AACR NextGen Grant for Transformative Cancer Research. The Northcott Lab is focused on solving molecular and cellular origins of medulloblastoma subgroups and their associated subtypes. Using a variety of highly integrative genomic and functional approaches, the Northcott Lab aims to elucidate the molecular mechanism responsible for medulloblastoma initiation, maintenance, progression, relapse, and metastasis. Novel candidate genes and pathways identified through these efforts are being functionally characterized in vitro and in vivo to improve the understanding of the biological basis of medulloblastoma pathogenesis and generate genetically accurate preclinical models. Moreover, we are applying our expertise in genomics and exploiting our unique access to large cohorts of patient material linked to prospective medulloblastoma protocols to contextualize the trial outcomes, identify novel clinically informative biomarkers, and develop innovative genomic analysis for monitoring disease status and predicting medulloblastoma relapse. The overarching goal of these efforts is to enhance the understanding of molecular and mechanistic basis of medulloblastoma such that we can improve the treatment and outcome for affected patients and their families. And with that, take it away, Paul.
All right, thank you, Amar, for a very kind and generous introduction. Hopefully everyone can hear me. Obviously, someone will notify me if there's any technical difficulties. I will tell you that this is surreal. Had anyone told me at the beginning of 2020 that my biggest talk of this year would be alone in my office, standing in front of my iMac, I would have told you were crazy. So these are definitely surreal times, and it's really an honor to be part of this Science of Childhood Cancer series that is hosted by the St. Jude Comprehensive Cancer Center. I am Paul Northcott, an associate member in the Department of Developmental Neurobiology here and affiliated with the Neurobiology and Brain Tumor program. Right, so for the last 15 years or more now, I've been studying this malignant childhood brain cancer known as medulloblastoma. This is one of the most common malignant childhood brain tumors, and it's diagnosed very early in life with a peak age of onset somewhere around 7 to 8 years of age, but it can also occur in very young infants, often less than a year into their first year of life, and certainly well into older adolescents and adults. And compared to other childhood brain cancers, especially of the malignant type, this is among the most common and most prevalent. And so there are various histological and demographic features that are well characterized in medulloblastoma. There is a sex bias towards a male preponderance, and this preponderance is associated with various genetic and molecular features, as I will describe throughout the course of my talk. The current standard of care for medulloblastoma involves attempts for gross total resection, craniospinal radiation in children older than the age of 3, and cytotoxic chemotherapies. And with these highly aggressive and often devastating treatments, there's obviously severe long-term consequences with treating young children with this very aggressive form of therapy. And even despite improved survival rates of maybe 60 to 80%, depending on the clinical risk of patients, there's still a great deal of improvement that's needed and an extensive amount of clinical heterogeneity. And so the reason I've stayed in medulloblastoma for so long and really focused on the single entity, I think, is best summarized in this slide here, where although all of these children look unfortunately very similar, and their tumors might look almost identical under the microscope, we know from studying their molecular landscapes, whether it be the genome, transcriptome, epigenome, or more recently proteome, that these are really a collection of different diseases that masquerade under the umbrella of medulloblastoma. And so it was during my PhD studies in Toronto and then later during my postdoc in Heidelberg that I really became fascinated by this molecular heterogeneity and fixated on trying to explain each and every patient that belongs or ascribes or affiliates with one of these core molecular subgroups. So as a community, we reached a consensus in 2012 of a 4 subgroup nomenclature, a 4 subgroup structure. And these include the Wnt, sonic hedgehog, group 3, and group subgroups. And these were first defined based on transcriptional profiling and more recently using more robust methods, including DNA methylation-based classification. And looking into the demographics, the histological variants, and the clinical outcomes, we quickly recognized as a community that there's utility in ascribing or defining medulloblastoma subgroup status. And you can see just looking at the survival data shown in this plot that there's a great deal of difference between a Wnt subgroup medulloblastoma and a group 3 medulloblastoma in terms of their progression-free or overall survival. So it's really this heterogeneity that's kept me going and kept me fascinated and fixated on this problem. And so for the course of this talk and in terms of the topics that I'd like to touch upon and highlight some of our key findings over the years, I think, our main goal is to make discovery. And I think what motivates me to come to the lab every day and to continue working on this pediatric brain tumors to make new discoveries, to better understand the biological basis of medulloblastoma, to better define the somatic genetic landscape so that we can interpret that molecular landscape in the context of developmental biology, tumor biology, and certainly in the context of clinical outcomes. And so these are just vignettes that I hope to highlight throughout the course of my talk, essentially all of which are derived from about 3 or 4 recent publications. And so starting with the first story, I really want to put my best foot forward and describe the genetic landscape of medulloblastoma and how we defined the recurrent somatic driver alterations according to molecular subgroup in a large cohort of over 500 medulloblastoma patients. And so this effort was born out of my postdoctoral research fellowship in Heidelberg, where I was fortunate enough to train under the mentorship of Stefan Pfister and Peter Lichter as part of the ICGC PED Brain Tumor Project, where we sequenced on the order of 250 plus medulloblastoma genomes and matched germlines. And we paired with Michael Taylor's lab in Toronto, who led the MAGIC Consortium, as well as the Pediatric Cancer Genome Project run out of St. Jude, and collaborators at the Broad Institute. to really define the genetic, epigenetic, and transcriptional landscape of medulloblastoma according to subgroup, so with the end game of trying to explain each and every patient. And so this is just a 10,000-foot perspective of the medulloblastoma genome in terms of recurrent driver gene alterations and genes that are either somatically altered or in rare occasions altered in the germline. And I hope what you can appreciate looking at this nearly 500 cancer genomes is there's a great deal of genetic heterogeneity. And when we divide these tumors according to molecular subgroup, you see specific thematic pathways emerge, such as activation of the Wnt signaling pathway in Wnt tumors, sonic hedgehog signaling in sonic hedgehog tumors, as you would expect. We also denote extensive deregulation of the chromatin and transcriptional machinery, and this occurs across all 4 subgroups, hitting different genes at different frequencies in different groups. But at the end of the day, we estimate that somewhere on the order of 50% of all medulloblastoma tumors have a clear somatic genetic hit in a chromatin modifying gene or a transcription factor, suggesting that a fundamental mechanism in the pathogenesis of medulloblastoma is deregulation of the epigenome and the transcriptome. So prior to this study, there had already been a first wave of next generation sequencing studies covering medulloblastoma, and these were conducted out of the Pediatric Cancer Genome Project here, the ICGC PedBrain Tumor Project in Heidelberg, as well as the Broad Institute. And those studies appeared in Nature in 2012, and results of which are summarized here in this bar plot. So it's not surprising that the majority of Wnt and sonic hedgehog tumors could be explained based on that first wave of sequencing papers due to the more highly recurrent genetic events targeting genes like beta-catenin, PATH1, SUFU that explain the Wnt in hedgehogs. But perhaps probably the most disappointing or sobering result was the fact that the group 3 and the group 4 tumors, which still have these generic names, were only partially explained. Only about a third of tumors from these particular subgroups could be explained by clear somatic driver events. And so in the current study, by leveraging a much larger cohort, nearly 500 patients, and looking beyond gene-level mutations, so looking beyond SNVs or indels that are targeting coding sequence, and beginning to look at non-coding genome space, taking into consideration copy number alterations and transcriptional changes, we have a much greater capacity to explain the group 3 and the group 4 tumors, somewhere on the order of 75 to 80% of those subgroups now explained based on somatic genetics. And I'm going to give you a couple of examples how we came to those numbers. And so this is just a very brief summary of recurrently altered genes in the group 3 and the group 4 subgroups. And this is not all of them. This is just a subset that are mutated at higher frequency. And you can see known oncogenes like MYC amplification dominating in group 3, again, amplification down in group 4. But we also begin to uncover new candidate genes. And this particular gene I want to highlight very briefly because it's one of potential interest in medulloblastoma and then also in a rare pineal tumor entity. So looking at the types of mutations that target KB2D4 in group 3 and 4, and I just should mention that these were identified at about equivalent frequency in both subgroups, we see a very striking pattern where the mutations are clustered very tightly in one particular domain of the protein. And what's also important to note here is that these mutations are not typical missense mutations or truncating variants, but rather in-frame insertions that result in the addition of 1 or 2 amino acids in this particular domain of the protein. And so looking into this protein in more detail, we realize that it's actually a very poorly characterized BTB back Kelch domain protein. And if you actually look in PubMed right now, you'll find our paper here, as well as the paper reporting the same pattern of mutations in pineal parenchymal tumors of intermediate differentiation, so a very rare and obscure pineal tumor entity, but that's likewise defined by the same type of mutation. And so looking into the function of other family members, this is a family of at least 30 proteins, we quickly realize that they function as adapters for ubiquitin ring ligases and essentially recruit substrates for protein turnover or protein degradation. And they do so through this beta propeller-like structure that's defined by the Kelch domain. And through homology modeling performed here at St. Jude, we were able to map the mutations, the hotspot mutations in KB2D4 to this one particular pocket that's presumed substrate binding surface of the Kelch domain. So although we don't understand the functionality of these mutations quite yet, we presume that they're disrupting physiological protein-protein interactions and somehow contributing to the pathogenesis of group 3 and 4. And so during my postdoc, I was very privileged to basically be drowning in whole genome sequencing data for medulloblastoma as a result of the ICGC effort. And as we showed in the first wave of sequencing studies, there was a very low frequency mutation rate in group 3 and group 4. There were few genes mutated beyond 10% of either subgroup. And so I was fixated on looking beyond gene level mutations and more looking into cytogenetic abnormalities or structural variants that might be playing a more prominent role. And I try to summarize one of the main findings that came from that analysis here, just by focusing efforts and focusing analyses on recurrent structural variants in group 3 and group 4. And so at the end of the day, we identified a locus on chromosome 9q34, shown here, where there was a series of different structural variants. So these included deletions, duplications, inversions, and more complex SVs that all converged on the same locus. And by layering in different data types, including chip sequencing data for active enhancer marks such as H3K27 acetyl, H3K9 acetyl, we see that the regions that are undergoing recurrent structural variation are characterized by these highly active enhancers or super enhancers. And as a result of these various classes of structural variation, what happens here is that these highly active super enhancers now become proximal to the normally dormant oncogene, in this case, GFI1b. So the GFI1b locus is normally repressed and marked by the repressive polycomb mark, H3K27 trimethyl. But as a result of the recurrent structural variations, we get mobilization of these highly active enhancers and now activation of the GFI1b promoter, leading to its overexpression. And so looking beyond GFI1b, it just so happens that it has a brother named GFI1 that maps to chromosome 1p. And there's a really funny story behind this that I would tell people offline someday. But basically looking beyond GFI1b, we realized that GFI1 was also targeted by recurrent translocations. Again, normally dormant, normally not expressed in medulloblastomas. But the recurrent translocations or intrachromosomal SVs brought the GFI1 locus proximal to highly active enhancers, super enhancers, leading to its activation. And so what we reported in this paper was that about 15 to 20% of all group 3 and group 4 medulloblastomas could be explained by this enhancer hijacking that leads to apparent activation of GFI1 and 1b. And so we wondered, based on the fact that these were serendipitous discoveries and never should have happened, we wondered if we could more systematically screen our medulloblastoma genomics data to look for additional examples or additional instances of enhancer hijacking. And so this was done in collaboration with Jan Korbel at the NBL, and specifically Joachim Weichenfeld in his lab, who's now got his own lab in Copenhagen, work with extensively. So Joachim developed a method known as CSAM, which was basically an unbiased pipeline for identifying enhancer hijacking events in cancer genomics data that requires sample match gene expression and copy number data as input. And so by applying this methodology to our medulloblastoma genomics data set here, this is the result. And so this is a QQ plot where anything above this line is potentially of interest and giving us signal. And so I just want to point out that we pick up the GFI1b locus, which is our positive control, using the CSAM methodology. But importantly, we uncover a new locus on 5q23 that appears to target a gene known as PRDM6. And notice if you actually follow these dots here, each one is a different gene, there are several hits on 5q23, including the SYNCAP locus. And so we looked at this locus in more detail, and I will tell you parenthetically, we had previously reported recurrent tandem duplications of the SYNCAP locus in group 4 medulloblastoma. About 20% of all group 4s have a clear tandem duplication that encompasses the gene. And so now looking at our whole genome sequencing data, we again replicate these tandem duplications, identify duplications that undergo inversions, and then also a series of more complex structural alterations. And then downstream here, about 600 kilobases is the PRDM6 locus. So how could the SYNCAP locus communicate with PRDM6, and how do these tandem duplications or more complex rearrangements lead to this aberrant induction of PRDM6? Well, first of all, if we look at the gene content at this locus, we see that there's a general gene dosage increase in samples harboring structural variation of SYNCAP. So you see SYNCAP expression here in SV positive case versus SV negative. There's a marginal increase in expression. Most of the other genes do not change in a significant manner other than PRDM6, which basically goes from off to on. And there's several log-fold increase in PRDM6 expression in cases harboring this underlying structural variation. And so jumping ahead to our model, and we have lots of data to support this, basically we determined that the SYNCAP locus itself is marked by a highly active enhancer or super enhancer, and it's normally contained within one chromatin loop or chromatin domain, or better known as topologically associated domain, shown here. And then in the neighboring TAD, the PRDM6 gene normally sits, and it's normally in an off position. But in these group 4 medulloblastomas harboring structural variation, we have a tandem duplication or a rearrangement that leads to copying of this super enhancer and repositioning of it in the neighboring TAD. So we get an additional copy of the SYNCAP locus along with its H3K27 acetyl, which then enables aberrant activation of PRDM6. And so those are some examples of kind of new cancer genes that we've implicated based on this approach. And now I want to talk a little bit about molecular classification and heterogeneity, intertumoral heterogeneity, so patient-to-patient heterogeneity. And, over the last several years, there's been a lot of evolution in terms of methodologies we can use to subgroup medulloblastomas and subtype them. Initially, these were gene expression platforms or gene panel-based expression platforms. But during my tenure in Heidelberg, there was really a revolution. And that was pioneered by Stefan Pfister, David Kapper, Volker Hofstadt, David Jones, who really embraced the ability of the DNA methylation-based classification approach to discriminate brain tumors according to underlying methylation signature. And this is an amazing resource, this paper published by Kapper et al. In 2018, where they basically introduced the use of the DNA methylation-based classification approach to over 2,800 brain tumors according to methylation signatures. Within that data set, you can see that the Wnt medulloblastomas have their own sort of discrete methylation profile, as do sonic hedgehogs in group 3 and group 4. And so we obviously wanted to dig into that more deeply in medulloblastoma. And so for this particular study, we had 1,256 patient tumors that we profiled on the 450K methylation array platform. You can see in blue here, the Wnt tumors are quite homogeneous, not a whole lot of substructure there, basically just a well-defined and homogeneous subgroup of medulloblastoma. Looking at sonic hedgehogs, we have quite some substructure and heterogeneity, and I'll talk about some of that as I go over the rest of my talk. But group 3 and group 4 is what I found the most fascinating, because just looking at this structure, it screams development, or it screams something that we totally don't understand at this rate. And so the spheres in yellow are the group 3 medulloblastomas, the spheres in green are the group 4s, and then somewhere along the midline here, you see this sort of mixing of samples. And so what we next did was we extracted the group 3 and group 4 tumors on their own. There's 740 patient tumors analyzed here. And when we break these apart, just focusing on the group 3 and group 4s, we start to appreciate additional substructure. And we define this substructure according to novel molecular subtypes, which we've designated as subtypes 1 through 8. As you can see, some are more homogeneous than others, and some blend into each other to some extent. And we've now validated these, and this was the original cohort of 740, and through meta-analyses and other large profiling efforts, we've looked at many thousand group 3 and group 4s as a community, and more or less come to this 8 subtype substructure. I just want to point out that when we look at these subtypes, we have some very clean subtypes. So subtypes 2, 3, and 4 shown here are all of group 3 parental origin, as shown in the plot on the right. Subtypes 8 and 6 at the bottom are 100% of the time group 4. But then we have this sort of ambiguity along the midline shown here, where subtypes 1, 5, and 7 really contain a mixture of group 3 and group 4. And I'll try to explain how we interpret that ambiguity when we get into the single cell analyses moving forward. And so just to convince you that the molecular subtypes of group 3 and 4 are genetically distinct, these are basically oncoprints summarizing recurrent genetic events by subtype. And just a few examples, we see subtype 1, which was a very homogeneous group. All of them have GFI1 or GFI1b enhancer hijacking. The subtypes 2 and 3 are enriched in MYC or MYC-N amplification. We see these oncogenic transcription factors amplified more frequently in subtypes 2 and 3. Subtype 4 is really a desert in terms of recurrent driver gene alterations. We don't have an explanation for that subtype. We definitely know these are malignant tumors, but we cannot explain them in terms of a putative driver event. And then jumping to subtype 8, this is an interesting subtype. It's the largest if you look at all group 3 and group 4s, and it contains a higher incidence or enrichment of chromatin modifier alterations. Genes like KDM6A, ZMIM3, MLL3, those are exquisitely mutated in subtype 8. And so, as I mentioned, we've come together as a community now and more or less reached a preliminary consensus or perspective on the subtypes of group 3 and group 4. And these are summarized here. And I will tell you that we've recently updated the World Health Organization classification of CNS tumors, and I'm thrilled and delighted to say that these subtypes are making their way into the next iteration of the WHO. So very gratifying to see that come to a level of recognition through WHO. And so you might ask, what do these subtypes mean clinically? And I'll just give you a couple of examples. And so by virtue of being here at St. Jude and working with world-class clinicians such as Dr. Gajar and Dr. Robinson, I've certainly had the privilege of profiling large clinical trial cohorts. This is exemplary data from SGMB03, a trial for newly diagnosed medulloblastoma patients that ran from the 2000s until just a few years ago and then was replaced by SGMB12, the current trial. This trial accrued 330 patients and then broke them down by clinical risk. And we've done molecular profiling on over 90% of this cohort. And you can see the subgroup breakdown for the average risk versus the high risk patients. And then likewise performed either whole exome or targeted sequencing for about 90% of this patient cohort. So we can look at mutational patterns, look at genes like P53, germline events, et cetera, that might be relevant in terms of clinical course. Looking at outcomes by molecular subgroup, this is for the average risk patients. We see the type of clinical heterogeneity we would expect, where Wnt patients do essentially universally well on this protocol. We have intermediate progression-free survivals for the group 4 and the sonic Hedgehog tumors, and then, as has been seen in many retrospective and more recently clinical trial series, the group 3s continue to do worse. However, when we break up the group 3 and the group 4s by subtypes, the subtypes I just spoke about earlier, we see, again, this extensive clinical heterogeneity in terms of progression-free survival. And so I draw your attention to subtype 3 here, which is essentially a cliff. These patients, as they come off therapy, unfortunately all relapse, or a very high proportion of them relapse, as you can see from this very steep decline in outcome. And so clearly we've identified a very high-risk subtype in subtype 3. On the other hand, we've also identified and extracted subtypes that do very well on this therapy, including subtype 7, shown in the light blue, and subtype 4 that I described earlier, where we don't know the genetic driver event. So I think that this is sort of the basis of the next generation of clinical trials. You will see these subtypes begin to become part of standard of care, and you will see in the next round of clinical trials run out of St. Jude that these subtypes will become upfront in terms of stratification. And so turning our efforts to infants now, this is a very important age group of medulloblastoma patients, and looking at molecular and clinical heterogeneity in a trial that again ran during the same era as SGMB03. This trial was known as SJYC07 and profiled 81 patients under the age of 5. And we know that the sonic hedgehog subgroup is very dominant in the infant population. So somewhere around 60 or 70% of infants belong to the sonic hedgehog subgroup. What was very interesting for us was when we applied DNA methylation array profiling, we were able to, in an unbiased fashion, discriminate the sonic hedgehog patients into two distinct molecular subtypes, which we designated subtype 1, shown in red, and subtype 2, shown in blue. Next, we looked at the clinical outcomes of these sort of unbiasedly discovered or retrieved subtypes, and we see clear difference in progression-free survival patterns where the subtype 2 patients do fairly well, around 80% surviving at 5 years, whereas the subtype 1 patients essentially do extremely poorly and relapse as they come off therapy. And so once again, I think this shows the utility of looking beyond the consensus subgroup status and getting into subtypes within subgroups, because there's clear biological and clinical heterogeneity within those subtypes. And so again, we're not the only ones to look at substructure in sonic hedgehog. There's been beautiful work done by Michael Taylor's lab and others in the field. And we've more or less kind of reached a perspective of a 4 subtype substructure for sonic hedgehog. I just spoke about the infant subtypes, which we're undergoing some sort of nomenclature evolution at the moment. We think that they'll be designated subtypes 1 to 4 at the end of the day. The sonic hedgehog alpha subtype is one I'm going to talk about momentarily when I get into the topic of germline predisposition. And so just a quick note on our efforts in relapse space. All of the work I've told you up till now is really only considering medulloblastoma diagnosis. So these are treatment-naive tumors that are surgically resected and then profiled by various modalities. And so a good friend of mine, Vijay Ramaswamy, had reported during his PhD that medulloblastoma subgroup status remains conserved between primary and relapse compartments. So if you're diagnosed as sonic hedgehog, you relapse as sonic hedgehog. If you're diagnosed as group 3, you'll relapse as a group 3. And so what we've done is collected nearly 130 paired primary and relapse samples and exposed them to molecular profiling, including the DNA methylation platform. And so in this alluvial plot, I think you can appreciate that in the vast majority of cases, that the subgroup status is exactly the same between the primary and the relapse compartments, as shown here, with some exceptions. And first and foremost, I want to draw your attention to the purple here on the right. So these are actually so-called relapses that were molecularly classified as high-grade glioma, and I'll talk about those momentarily. So roughly about 10% of our so-called relapses were redefined in this study based on molecular classification using the methylation array. And we also see a subset of subgroup switching between group 3 and group 4, and I just want to highlight that here. And so we have 4 primary tumors diagnosed with a high confidence group 4 based on DNA methylation that are then recurring as high confidence group 3, shown here. And one exception where we have a high confidence group 3 that then relapses more as an intermediate group 3-4, as highlighted in this case. And when we look at the genetics underlying these rare subgroup switching cases, we see, although somewhat anecdotal, a common theme, and that is genetic events targeting MYC, MYCN, or FBXW7 that emerge at relapse. And we know FBXW7 plays a critical role in the regulation of MYC and MYCN. So although it's really underpowered to come to any robust conclusions, we think that there's a possible mechanism whereby activation of MYC or MYCN might drive this subgroup switching from group 4 to group 3. And then just a quick note on the secondary tumors that we've identified. We see that the primary tumors span multiple subgroups. We have Wnt, sonic hedgehog group 3 and sonic hedgehog group 4 primaries that are then recurring, so to speak, as subsequent high-grade gliomas. And when we actually look at both the clinical details as well as the genetics of these tumors, I think we fully substantiate these findings. So this is the genome-wide CNV plot derived from methylation for the group 4 primary tumor, or one of the group 4 primary tumors in this case. We have an isochromosome 17, like a very hallmark feature of group 4 medullo. And then the emergence of glioma-specific oncogenic events, including c-Met amplification, CDKN2A2B homozygous deletion. And so just to summarize this first half of my talk, I hope that I've convinced you that by profiling a very large cohort of medulloblastomas, we've gained a better understanding of the genetic events driving each and every subgroup and now explain a much higher proportion than previously understood. And we've implicated novel cancer genes in the process, including KBTBD4, PRDM6, and others. By profiling the methylome of over 1,200 medullos, we see a higher degree of substructure than previously anticipated, dividing core subgroups into multiple subtypes, and shown clinical relevance of these subtypes in prospective clinical trial cohorts. And then just in that last vignette, I think I've shown you in our studies of primary and recurrent medulloblastoma that there's rare subgroup switching that occurs most commonly between group 3 and group 4, and that roughly 10% of so-called medulloblastoma relapses were molecularly reclassified as high-grade glioma. And so next I want to move on to genetic predisposition. So this is a very important topic when we're considering infants and young children who have really not been exposed to the same type of insults that promote adult malignancies, right? So we had long thought that there was a larger proportion of genetic predisposition that we just didn't know about or that we just didn't completely understand. And by taking advantage of the large cohorts of germline sequencing and matched tumor sequencing, we next sought to look at the incidence of cancer predisposition across this very large cohort of over 1,000 medulloblastoma germlines. And we've initially focused our analysis on 110 cancer predisposition genes. And in doing so, it's not surprising we were able to identify some of the usual suspects, including recurrent mutations in SUFU, PATCH1, and P53, all in the germline of sonic hedgehog medulloblastoma patients. We identified about 10% of Wnt patients that had germline APC mutation, usually in the absence of somatic beta-catenin, and then lower frequency mutations in DNA repair genes that occurred across subgroups targeting PALB2 and BRCA2. And so at the end of the day, based on these 110 cancer predisposition genes, we estimated that roughly 6% of all medulloblastomas have a clear pathogenic hit in one of these known cancer genes. And this frequency and incidence varies by subgroup, with the highest frequency of cancer predisposition predicted in sonic hedgehog. And so I can tell you, as someone who's very motivated by discovery and trying to identify new genes and really kind of new insights into disease biology, although I think this was an important effort, it certainly didn't cause my motivation to catch fire, because at the end of the day, we want to identify something new. And so fortunately, we kept working at this, and Stefan, I have to give plenty of credit to him and the group at EMBL for continuing to dive into this data set and look beyond the usual suspects. And so in this particular approach, we decided to throw away the 110 cancer predisposition genes and now screen genome-wide, looking at all protein coding genes for pathogenic germline variants that were protein truncating in nature. And so not surprisingly, as shown in the Manhattan plot here, we identify our favorite predisposition genes, PATCH1, SUFU, and P53, but it would be impossible to overlook this signal on chromosome 9q, targeting a gene known as IKB-cap, or better known as ELP1, or elongator complex protein 1. And so we quickly looked into ELP1 function and realized that it's a highly conserved scaffolding subunit of this complex known as elongator. It's a 6 subunit complex, and ELP1 functions to hold it together. And so again, looking through the literature, we quickly realized there had been multiple functionality ascribed to elongator, but the most important or the most relevant seemed to be its importance in modifying transfer RNAs, and specifically adding carboxymethyl groups to the uridine 34 position of the anticodon loop of tRNAs. And what these modifications do is they enable efficient translational elongation and flexibility with respect to Watson-Crick base pairing, so allowing for flexibility of either AA or AG ending codons. And so looking at our medulloblastoma data set in more detail, we realized that all of these ELP1 germline mutations were restricted to sonic hedgehog patients, and about 10% of all sonic hedgehog patients have a clear pathogenic germline variant in ELP1. Next, we break hedgehog up by age, and we see that it's actually very commonly identified in childhood patients, very rare in infants and not found in the adults. And importantly, when we compare ELP1 germline mutations to other well-recognized tumor suppressors and predisposition genes, we see that it's more than double the frequency compared to something like SUFU, P53, or patch. So we quickly got on board and realized that this was an important finding. And so working with Kim Nichols and Kayla Hamilton in the Division of Cancer Predisposition here, we started to look at our St. Jude patients and our St. Jude families. And this is a very important family to feature, where we have a remarkable familial transmission of the ELP1 germline variant. And so looking at the proband here, this is a 4-year-old boy who was diagnosed with desmoplastic medulloblastoma, carrying a pathogenic ELP1 mutation. By performing parental sequencing of both the mother and father, we quickly realized that this individual inherited the pathogenic ELP1 allele from his father, who remarkably was also diagnosed with medulloblastoma as a teenager and later developed colorectal cancer. Looking at the paternal aunt, we see medulloblastoma diagnosed during childhood and an unfortunate death early in life, and then multiple additional brain tumors of not otherwise specified diagnosis identified on the paternal side. I think this quite convincingly shows that this is functioning as a bona fide cancer predisposition gene and being transmitted across generations. And so looking at the pattern of mutations, we quickly identified the mutational pattern, and that these mutations span the entire coding sequence, and they're always nonsense mutations, frame shift mutations, things that would result in clear loss of function. And screening the medulloblastoma germline, we realized that the mutations are heterozygous in the germline. And then looking at the associated tumor genomes, we quickly came to the conclusion that 100% of ELP1 germline carriers undergo 9q loss of heterozygosity in their associated tumor genome. So this is, again, classical features of a tumor suppressor gene being inactivated. So next, we leveraged our large cohort of sonic hedgehog medulloblastoma sequencing data. This is over 200 patients where we've got whole genome, germline, and tumor sequencing. Here are the ELP1 mutations, all falling into the sonic hedgehog alpha subtype, shown here. And what is known about this subtype is this is the subtype of childhood, so that makes sense. And we see more or less mutual exclusivity with events targeting P53. And I think that's better visualized here. When we specifically look at cooperating events in ELP1 tumor genomes, we see a very high incidence of patch mutations. About 80% of these genomes harbor inactivation of patch, and a lower frequency of PPM1D and MDM4 amplification. On the flip side, we see complete mutual exclusivity with P53, germline or somatic mutations, as well as MECAN and GLE2, other potent oncogenes in this subtype. And so next, working with Olivier Rowe at the Curie Institute, we leveraged our access to quantitative proteomic data and compared ELP1 mutant versus ELP1 wild-type proteome. And as shown in the volcano plot, there's a clear bias towards upregulated proteins in the ELP1 mutant tumor. So I don't know, I can't remember the exact numbers, but several-fold increase in the number of proteins being upregulated versus downregulated. And you'll quickly notice that elongator subunits, including ELP1 and ELP3, which is the catalytic subunit of elongator, are highly and significantly downregulated in the ELP1 mutant proteomes. Functionally, in terms of functional annotation of the differentially expressed proteins, we see upregulation of proteins involved in amino acid activation, protein folding, and other pathways that we would presume to be related to elongator dysfunction. And we see downregulation of genes involved in neuronal differentiation. So next, to put the nail in the coffin, if you will, and really get to the heart of the mechanism, we teamed up with Brandon Wainwright at Queensland University, who's an expert in elongator, as well as sonic hedgehog medulloblastoma, coincidentally. And he had experience profiling transfer RNAs and specifically quantifying tRNA modifications. And by providing Brandon with ELP1 wild type and ELP1 mutant sonic hedgehog tumors, he was able to show us quite convincingly that there's a severe downregulation of the elongator-dependent tRNA modifications in the ELP1 mutant tumors compared to wild type. And here is data for 2 of those elongator-dependent tRNA modifications. And so for the summary of the second part of my talk, now I hope I've convinced you that genetic predisposition to medulloblastoma is important. Our initial screen estimated that anywhere between 5 to 6% of all medulloblastomas could be explained by pathogenic germline variants in 6 different genes, and that risk varied according to molecular subgroup, with the highest risk seen in Sonic-Ketchuk patients. But I put asterisks beside these numbers because we really have to throw these numbers out now. By extending our search space beyond known hallmark cancer predisposition genes, we've uncovered ALP1, which is mutated in 20% of childhood Sonic-Ketchuk medulloblastomas. So this is doubling this now from 20 to 40% just by adding in one additional gene. Hopefully I've convinced you that the ALP1 germline variants are heritable. They're restricted to the Sonic-Ketchuk alpha subtype. We've observed universal 9-QOH and somatic inactivation of patch1, so we're getting a really good understanding of the tumor biology. And then by really digging deep into mechanism, we've shown that ALP1 mutant tumors exhibit elongator deficiency, tRNA dysfunction, and translational reprogramming. And so in the last 6 or 7 minutes here, I'm going to just switch gears now and get beyond bulk tumor genomes and into the weeds of single cell genomics. And so this is a study that we published last year, working together with Mario Suva, Mariella Philbin, Brad Bernstein, and Volker Hofstadt, all of whom are in Boston, to really get into intratumoral heterogeneity in medulloblastoma. And by taking freshly isolated medulloblastomas seen on medulloblastoma clinical trials run here at St. Jude and through our collaborating sites, we were able to put together a collection of 25 freshly resected tumors and 11 medulloblastoma PDX for profiling on the SmartSeq2 platform. So this is the single cell RNA sequencing using a very deep plate-based profiling approach, sequencing a total of 8,500 malignant cells and quantifying upwards of 5,000 genes per cell. So looking at this data set here, these are all our tumor cells colored according to the subgroup from the bulk tumor which they were derived. We see that Wnt tumor cells grouped together as shown here in the top left, Sonic-Ketchuk tumor cells grouped together according to patient in the top right, and then we see the group 3 and the group 4s here. In purple, we basically are analyzing the patient-derived xenografts, the majority of which are MYC amplified, so they're a lot more similar to each other. One thing to mention in our FACS and plate-based approach, there was very little immune infiltrate or normal cell contaminants, so highly malignant population that we're able to verify by mutations and copy number variants. And so just for the sake of time, I only want to show you one main story here. We looked specifically at intratumor and cellular programs in the group 3 and the group 4 medulloblastomas. And so here, by taking nearly 5,000 malignant cells from group 3 and group 4 and doing a pairwise correlation analysis to look at similarity, there are a few take-homes. One, each individual tumor is more similar to itself than to its neighbor, as you can see by the deep red here. And this could be explained by underlying copy number variation or mutations. But by and large, the group 3 tumor cells look more similar to each other. The group 4 tumor cells look more similar to each other. And then we have a subset of cells here that exhibit features of both group 3 and group 4. And by performing various bioinformatic approaches and pipelines, we define 3 different MET programs or expression programs in these malignant cells, including a mitotic A program, undifferentiated progenitor-like program, which we designate the B program, and a more differentiated neuronal-like program, which we designate the C program. And I'll walk you through a couple of examples here to show you what I mean. And so looking at a prototypical group 3 medulloblastoma, every column here is a single cell, and we've quantified the expression of either the undifferentiated program in B or the more differentiated program in C. And you can see in this prototypic group 3 that the vast majority of cells are exhibiting expression of the undifferentiated progenitor-like program. And really only a subset of cells are positive for the differentiation program. On the other hand, when we look at a prototypic group 4, it's the complete opposite, where there's clear lack of the progenitor-like program and a much more dominant differentiation associated program shown here. And then just to muddy the waters, we look at a group 3-4 intermediate, one of those bulk tumors that would straddle the midline of the group 3-4 subtypes, and we see features of both the undifferentiated program as well as the more differentiated program shown here. And so we think now we have a cellular explanation for this ambiguity between group 3 and group 4, where depending on the degree of the differentiation state, you either classify as a group 3, a group 4, or an intermediate. And so by projecting these programs into bulk expression data, I think we just visualize more or less the same pattern, and that is the group 3 tumors are more or less defined by the undifferentiated progenitor-like program, the group 4s by the more differentiated neuronal-like program, and then a subset of about 15 to 20% that exhibit the expression patterns of both programs. And again, that data is shown here in the heat map. And what I think was quite gratifying is that when we sort these bulk tumor samples based on these underlying differentiation programs, we group all the group 3s on the left, all the group 4s on the right, and then we have about 15 to 20% that kind of sit in the middle here, which are mixed classification based on DNA methylation. And so next I want to close with our insight into developmental origin. And so in parallel to our medulloblastoma single cell sequencing studies, we performed an unbiased analysis of cerebellar development using the 10x DropSeq platform, sequencing 13 developmental time points and generating single-cell RNA-seq data for nearly 90,000 cells, and then interpreting those cells in the context of populations, and essentially replicate cerebellar development as shown by the 18 different cell types highlighted in this t-SNE plot. And so one of our main goals here was to integrate or compare our normal cerebellar atlas to our malignant single cell data set in an attempt to explain the identity of the malignant cells and likewise for putative subgroup or subtype specific cellular origins. And so just to give you the punchline of this data, by comparing the 18 different cell types in the normal mouse cerebellum to our medulloblastoma data sets, whether they be bulk or single cell, the greatest correlation that we see for sonic hedgehog medulloblastoma is the granule neuron lineage, or GNPs, shown here. This would be expected. The cell of origin for sonic hedgehog is well defined as being progenitors within the granule neuron lineage. But the new discovery here was implicating UBCs, or unipolar brush cells, and glutamatergic cerebellar nuclei, two glutamatergic populations that are born out of the upper rhombic lip, shown here as UBCs, and glutacn here as being highly correlated with group 4 medulloblastoma. And so this was really the new insight to come out of this analysis. And in parallel, my former PhD supervisor, Michael Taylor, performed a very similar analysis, again, recreating cerebellar development in the mouse and comparing that to medulloblastoma single cell and bulk expression data sets, coming to the same conclusion. So completely independent effort, but coming to the same point of convergence, and that is high degree of similarity between group 4 and the UBC lineage. And so just to quickly summarize our interest now, obviously, if group 4 medulloblastomas are highly similar to either glutamatergic cerebellar nuclei or unipolar brush cells, we want to know more about those populations. And we've used insights gleaned from our single cell RNA sequencing data set to identify biomarkers of these populations, and now engaged in lineage tracing approaches to enrich for these populations, so we can either study them on a much deeper level, or we can perform somatic perturbation or germline perturbation in these particular populations at the right time point with the right candidate genes. And so in summary of this last part of my talk, I hope that I've been able to convince you that bulk medulloblastomas exhibits specific cellular hierarchies, and we now have a much better explanation of group 3 and group 4 and the so-called intermediates that lie in between. We've demonstrated that single-cell RNA sequencing recapitulates cerebellar development and informs medulloblastoma cell types, and likewise implicated the unipolar brush cell lineage as well as the glutamatergic cerebellar nuclei lineage as candidate group 4 medulloblastoma cells of origin. And so none of this work would have been possible without team science. And I've been a big proponent of team science since my early days in this business. And I've had the opportunity to work with some of the best people in the world. And many of them are included on this slide. These are sort of the primary external collaborators that I've worked with throughout the course of my career. And I'll just mention that these large profiling efforts where we've literally screened thousands of tumors would not be possible if we didn't come together as a community and we didn't leverage expertise, insights, access to clinical trials, clinical data, techniques that are only available at one institution or better at another institution. And so I owe a lot of gratitude to all of the primary external collaborators shown on this slide. And then likewise here at home, I have the great privilege of coming to work every day with some of the best clinician scientists in the world with respect to medulloblastima, as well as some of the best basic scientists focused on this disease. And a special shout out to Giles Robinson, who's we're sort of like shake and bake when it comes to studying this disease. And so I'm very grateful for our team effort to try and solve this problem of medulloblastoma. And then finally, all of the funding agencies that contribute to these big, large-scale studies. I have to mention support from the Brain Tumor Charity, St. Baldrick's Foundation, the Sontig Foundation, the Pew Charitable Trust, my lab that gives me motivation for coming into work every day. I will mention that we're recruiting at all levels, ranging from scientific manager to bioinformatician to postdoc to graduate students. So please feel free to contact me and send me your CV if you're interested. And I think we are out of time, but I'm certainly happy to stick around for questions. And thank you for attending today.
Thank you, Paul. We've got a couple of minutes before the top of the hour. So I think we can ask you one question, and then you're welcome to stay on for more questions. So the question is, could you comment on the single ELF1 mutation case for sonic hedgehog gamma subtype? Do you think this indicates ELF1 mutations can occur in other subtypes, or that this case may be mis-subtyped?
Yeah, that's a great question. So I can tell you we looked very closely at that exception, and we were quite confident with the subtype classification. It just doesn't obey the rule. And I have to look back at the age of that patient. It's probably a younger patient. It's just straddling sort of the border between the infant and the childhood subtype. So we're confident with the subtype assignment. And it's just, that's the nature of science. Not if everything obeys the rules every time, then there's something wrong with your data probably.
Okay, so one more and then we'll stop. So this is from John Lucas. A figure was presented at the COG meeting, which was a reanalysis of the ACNS 0332 data, which showed a subgroup-specific benefit of CSI plus carboplatin in group 3 patients. Is it going to be possible to assign these patients to the right subgroup for assigning therapy in real-time fashion without methylation? Will there be IHC classifier similar to what is used for SGMB12, which is practical and usable for those without access to advanced high-throughput molecular analytical techniques?
Okay, there's a lot in that question. I can tell you that, I'm obviously a huge proponent of the DNA methylation-based classification. There are many institutions in Europe and now here in the US and North America that are applying the methylation array clinically, and St. Jude is on the cusp of that. And so I would absolutely envision that the next round of clinical trials, certainly out of St. will be incorporating the molecular classification and subtyping based on DNA methylation. I don't think, unfortunately, based on our experience with IHC, that will be a reliable approach to subgrouping or subtype. We've certainly tried that over the decades with marginal success. Some subtypes or subgroups have clear biomarkers such as Wnt and sonic hedgehog, but group 3 and 4 for a reliable classification, we need to use methylation array.
Okay, it's the top of the hour, so I'd like to thank Paul for his excellent talk and close the official event, and also invite all those who are interested to stay on for a continued discussion and further questions that you may have. Thank you all for coming, and I encourage you to visit the website and mark your calendars for the other upcoming talks, including Crystal Mackles from Stanford University next Thursday, speaking on CAR T cells for pediatric cancers, and Rick Young and Isaac Klein on MIT and Dana-Farber Institute on October 22nd, speaking on partitioning of cancer therapeutics in nuclear condensates. So with that, those who want to stay on, Paul is generously donated.
Happy to stand here for 20 or 30 minutes. So if there are questions, then please.
So a couple of questions. This is from Beth Lawler. Uh, it is interesting that the KMS curves show very rapid progression for high-risk patients right as they come off therapy. This suggests that they have not developed drug resistance, but have instead had their disease controlled effectively by the therapy. How do you think this should influence the approach to therapy in the next trials? In that absence of new targeted therapy, should we consider adding maintenance course of therapy for these high-risk patients?
Right. That's an excellent question, and I think one better suited for Giles or you, Amar. But I'm happy to comment, at least from my perspective. So, if we look at the data from SGMB03, and we see subtypes like subtype 3 that are declining immediately as those patients come off therapy. So those patients are obviously going to need to be grouped into their own high-risk stratification arm of the next clinical trial. And then likewise, we'll look at subtypes like subtype 7 and subtype 4 and think about putting them in a low-risk arm and de-escalating therapy. So the question is, what do you do? How do we treat those high-risk subtypes and those patients that are essentially relapsing as they come off therapy? And so I think, that's probably not the best question for me. I can tell you right now there's a paucity of targeted therapies that are showing any efficacy, especially in the group 3 and the group 4 patients. We have some internal experience now with Giles Robinson's St. Jude Dawn trial looking at cyclin-dependent kinase inhibitor, et cetera, but it's still too early to report whether or not that would be something that could be given up front to these more high-risk patients as an adjunct to standard high-risk therapy. But obviously, we have our eyes on those key players and those bad performing subgroups and subtypes, and are currently strategizing how we're going to approach them in the SJMB22 trial as SJMB12 begins to wind down.
Okay, this one, Paul. For example, with the tandem duplication of SYNCAP locus, do you understand why the duplicated locus ended up in the next loop over instead of extruding out of the source loop? Also, do you see overexpression of SYNCAP in that instance to support your model?
Okay, so so the model that I presented was based on our data from 2017, right? So we actually put that paper together in 2016, and the model was assembled based on our perspective then. And what I didn't show you was all the data that kind of provided that perspective. But essentially, we know that predominantly the SYNCAP locus, which is highly expressed, marked by a highly active enhancer or super enhancer, usually undergoes a tandem duplication. Sometimes that duplication can involve an inversion or more complex rearrangement. Basically by overlaying TAD domain structure at that time, not from medulloblastomas, but from ES cells and fibroblasts. But we know TADs are highly conserved across cell types and tissues. We basically were able to demarcate the TAD boundary between the TAD which contains SYNCAP and the TAD which contains PRDM6. And we also had CTCF ChIP-seq and H3K27 acetyl ChIP-seq. And so the model was basically, what we presumed to be the mechanism of aberrant activation. So more recently now, again, carrying this work forward, we've done high-C, capture-C, and more, really drilled down on the mechanism. And, all I can, that data is not in a presentable format right now, but I'd say that we've more or less verified the model that we've proposed in 2017, now using the chromosome conformation-based assays.
Okay, any more questions from the panelists? This is from Madan Babu. Great talk and inspiring work, Paul. When you analyzed your cancer predisposition variants in families, did you only consider protein coding regions or did you consider non-coding variants? Did you consider methylation pattern in the predisposition genes to infer whether the non-coding variants affect expression?
Okay, so the short answer is no. We basically, when we expanded the search space, and even the original search space, which was the 110 cancer predisposition genes, we were really only looking for highly deleterious mutations that affect the coding sequence. And these would be frame shift, nonsense, splice site types of mutations. We did not, the exception being P53, which often has missense mutations in the DNA binding domain. So when we expanded the search space to include all protein coding genes, we again focused on the highly deleterious alleles, and we did not look at promoter mutations or enhancer mutations or DNA methylation patterns. That's something that I think we'd love to explore, as we continue to try and explain additional families that we can't currently explain based on the genes we've identified thus far. So I hope that answers your question.
Okay, so I think that's it. Thank you, Paul. Fantastic
No problem.
talk. And thank you, the panelists.
Thank you.
Thank you.